All right, so we're going to pick up here. We've been talking about the adaptive immune system, and now we're going to look specifically at humoral immune response. So remember, humoral immune response is going to involve B lymphocytes or B cells. So whenever a B cell does encounter a target antigen, it's going to provoke a humoral immune response. At that point, antibodies for that particular antigen are going to be produced. The B cells, once again, they're activated whenever antigens bind to those surface receptors. This goes back to if the previous video we talked about once the antigen binds, it's going to lead to the proliferation and the differentiation of B cells into effector cells. The vast majority of the clone cells are going to become plasma cells, and plasma cells are antibody secreting effector cells. And they're going to secrete a specific antibody or specific antibodies at a rate of about 2,000 or so molecules per second. And this is going to last for 4 to 5 days, and then the plasma cells will die. The antibodies will circulate around in the blood or the lymph, and whenever they encounter a free antigen, they will bind to it, and this is going to mark it for destruction by either innate or other adaptive mechanisms. Now, like I said, the vast majority of the clone cells become plasma cells, but the ones that don't become memory cells. These are going to provide immunological memory. And what this is going to allow is it's going to allow for our immune system to mount an immediate response to future exposures to that same antigen. So this is just showing that we have cursor, here we go. Antigen binds to lymphocyte. What I want to point out here is that most of these are going to be plasma cells, a few are memory cells. Kind of ignore the writing. All I wanted to point out was that we have more plasma cells than we have memory cells. So remember, I just talked about memory cells, and we're now going to transition into immunological memory. So again, the shortcoming of the adaptive immune system is the fact that it requires priming. We have what's called the primary immune response, and this is where there is cell proliferation and differentiation when the lymphocytes exposed to the antigen for the first time. There's a lag period, 3 to 6 days. Peak antibody levels are going to be reached at about 10 days, and then at that point, antibodies levels decline. Secondary immune response is from second and subsequent exposure to the same antigen. When these subsequent exposures to the same antigen occur, the immune system has a more prolonged, more effective response. Those memory cells are going to provide immunological memory. So instead of responding within days, at 3 to 6 days, they will respond within hours. And antibody levels are going to peak in 2 to 3 days instead of the 10 days. And on top of that, when they peak at much higher levels. Antibodies will bind with greater affinity, and the antibody level can remain high for weeks to months. So what we're looking at here is this would be exposure to antigen A. This is day 0. You can say it takes about, on this graph, maybe 5 days to respond. And then this is looking at antibody level. It's going to peak at about 10 days after the exposure, and then it's going to go down. And let's say that the individual on this day, so 4 weeks later, is exposed to the same sorry about that, is exposed to the same antigen. We see a much more rapid increase in antibody level, quicker, and it goes up a lot higher, right? So it goes up way up here, and then it doesn't taper off nearly as much. So within 10 days to, this is 18 days, it's already down to almost zero. And here we have, this is 4 weeks and it still hasn't tapered off. So the take home: secondary humoral immune responses, more rapid, uh, and on top of that, produce more antibody faster, and the antibody will actually bind with greater affinity. All right, so now we move on to talking about active and passive humoral immunity. So, active humoral immunity is going to occur when B cells encounter antigens and produce specific antibodies against them. There are two types of active humoral immunity: there's naturally acquired and artificially acquired. Naturally acquired active humoral immunity is going to be formed in response to actual infections, either bacterial or viral, but the individual has to have gotten the infection themselves. Artificially acquired is going to be formed in response to a vaccine of dead or attenuated pathogens. And attenuated pathogens this just means the pathogen there's a reduction in the virulence or the harmfulness of the pathogen. And what vaccines do is they just spare us the symptoms of the primary response. So if we go back, what the vaccine is going to do is the vaccine is going to allow this to kind of happen, but because there's less virulence, we're not going to respond, we're not going to get us sick. And then if we're exposed to the same thing a second time, you can see our immune system is going to react much more rapidly. Then we have passive humoral immunity. And this is going to occur when ready-made antibodies are introduced into the body. That's why it's passive. So the B cells themselves, again, this is passive, are not challenged by antigens. As a result, immunological memory does not occur. Protection ends when the antibodies degrade. That again is because of the fact that no memory cells are being made. Okay, so as soon as those antibodies degrade, it's over. There's two types of passive humoral immunity: naturally acquired and artificially acquired. Naturally acquired would be antibodies delivered to a fetus via placenta or to an infant through milk. And then artificially acquired would be injection of serum like such as gamma globulin. Cannot speak. So the protection is immediate, but it's going to end again whenever the antibodies naturally degrade in the body. And so this is just a nice little kind of chart of the different types of humoral immunity. And I'm going to stop here. I'm going to do one more video about antibodies.